Hey everybody, I was driving home last night and decided that we had really cruised through the blood chapter pretty quickly, and I just wanted to take an hour or so to kind of recap and go back over it. If you are already feeling like you have a pretty good understanding of it, don't feel like you need to listen to this. I'm not going to cover anything new. I'm just going to go back through and kind of highlight some of the key points and review some of the stuff that seemed pretty confusing last night. All right, so I'm going to go right into our first slide on blood. Blood is the only fluid tissue in our body. It's also a connective tissue. That's what I really want you guys to focus on, is that blood is a connective tissue, and I want you to be able to understand why it is a connective tissue. The main reason is that it has both cellular and liquid components, like connective tissue. There are cells living in the connective tissue surrounded by an extracellular fluid-like matrix. And that's what we see in blood. Blood's, like I said here, a highly specialized connective tissue composed of formed elements, which are cells and platelets, red blood cells, white blood cells, platelets, suspended in a fluid matrix, a fluid extracellular matrix that we call plasma. I guess the big difference between blood and other types of connective tissue is the absence of collagen, and elastic and reticular fibers, and that's why blood is fluid and not more of a gel-like material like our connective tissue, because it's lacking those fibers. Blood does, however, have dissolved proteins that during the clotting process can actually turn into fibers, so it is very similar to connective tissue in that sense. It's just missing those fibers. So if you compare that to areolar connective tissue and dense regular connective tissue that relied heavily on these different types of fibers for its structure and support, blood doesn't have that. It does have formed elements, cells, and an extracellular fluid matrix, and that's why we call it a connective tissue. All right, so if we take a sample of blood out of someone, that dark red, thick blood that comes out of someone, that's whole blood. That's all the different components of blood in solution, mixed up blood, whole blood. But if we go ahead and centrifuge that blood sample, which means basically spin it very quickly, all the more dense elements of the blood are going to settle on the bottom, and then some of the less dense elements will settle on top of that, with the least dense part of blood floating on the top. And that's what we see here in the test tube. The red on the bottom is made up of erythrocytes. The small white layer, called the buffy coat, is made up of leukocytes and platelets. And then everything on top of that, the orangish color, is blood plasma, the fluid component of blood, the fluid extracellular matrix of blood. So erythrocytes, again, that's the bottom. Those are the red blood cells. This is the oxygen-carrying portion of blood. This is referred to as our hematocrit. The hematocrit is our oxygen-carrying portion of our blood, and it's composed of erythrocytes. And this accounts for about 45% of our whole blood. Okay, leukocytes and platelets make up less than 1% of whole blood, and they comprise the small, thin, buffy coat in between the erythrocyte layer, the hematocrit, and the plasma, which again is the orange. And this plasma comprises about 55% of whole blood. These numbers can vary a small amount. I'm sure there'll be more leukocytes in circulation. If there's a large infection, there'll probably be less plasma if we're very dehydrated. But as a general rule, we can remember these numbers, and that each one of these components typically constitutes a certain percent of our whole blood. Functions of blood. First and foremost, blood acts to distribute things throughout our body. Distributes gases like oxygen and CO2, nutrients picked up in our digestive system, for example, or glucose brought from the liver to the cells of the body, hormones like we learned about last week, released by endocrine glands. Blood is a means to distribute things throughout our body. That's why it's so important that it's fluid. It's a fluid tissue. I'm going to go ahead and just read here the distribution functions of blood. We pretty much covered them, but I want to read from the book. Okay, so blood delivers oxygen from the lungs to the body and nutrients from the digestive tract to all body cells. blood transports metabolic waste products from cells to elimination sites. So an example being carbon dioxide produced in our body's tissues is transported to our lungs to be removed from the body. We can transport waste, different types of nitrogen waste, uric acid, things like this, to the kidneys for elimination, and then transport various other wastes to different elimination sites. None of those specifics do you really need to know, other than later we'll talk a little bit about how oxygen and carbon dioxide are transported by blood. The second big category of blood function is that it maintains temperature, maintains pH of our body, and that it maintains our fluid volume. So it maintains appropriate body temperature basically by regulating how much blood is, let me rephrase that. It's going to absorb heat throughout the body and bring that to the skin surface to encourage heat loss. And then our vascular system can actually reduce the amount of blood transported to our skin in order to promote heat retention. Okay, so blood plays an important role in both releasing heat from our body and maintaining heat within our body. Blood is also going to maintain normal pH in body tissues. Many of the proteins and different types of solutes in our blood act as buffers. Buffers prevent wild swings in pH, whether a swing to become more acidic or basic. And then pH definitely plays a big role in all of our body functions, body chemistry, our chemical cascades, things like that. So it's very important that blood maintains a steady pH in order for all of our cells to function properly. Blood proteins in the blood, and we talked about this a little bit last night, specifically albumin, are going to maintain our fluid volume. They prevent loss of fluid from the bloodstream into the tissue. This is important because if our fluid all leaked out into our tissues very quickly, our blood pressure would drop, our fluid volume would drop, and our tissues would be saturated with fluid, and the cardiovascular system would not be able to perform its other functions adequately, like distribution, for example. Lastly, blood acts to protect our body. It protects our body from blood loss, and we'll talk about that when we look at clotting. When a blood vessel is damaged and blood is leaking from that blood vessel, it can quickly clot and stop that leak. Blood also helps prevent infection. Scattered throughout blood are antibodies. Remember, antibodies destroy foreign cells by binding to antigens, and we'll talk a little bit about a specific type of that later when we talk about blood type. Also, white blood cells circulate throughout the blood, and they phagocytize bacteria, release chemicals to destroy bacteria, chemicals to destroy parasites, things like that. So blood plays a very active role in protection, especially in immunity. Okay, let's go ahead and move on to blood plasma. Blood plasma is the largest component of blood. It represents about 55% of whole blood, and blood plasma itself is 90% water. Let's see. Blood plasma is very important because it is what makes our blood fluid, and it's also a solution for different types of things, nutrients, gases, hormones, proteins, ions, different types of wastes. It's a solution for those things to be dissolved into and to be transported throughout the body. Now, the one I really want to focus on in terms of testing and things is albumin. Albumin comprises the bulk of plasma proteins, and it maintains our plasma's osmotic pressure. All right. So albumin actually, it likes to bind to water. It likes to grab water and hold on to water, as well as some other types of ions. And because there's albumin in our blood plasma, water is attracted to blood. Water is attracted to albumin, so therefore water is attracted to our blood. If that wasn't the case, remember, water can easily leave a capillary if it's not for a gradient, an osmotic gradient. So when we get out into the periphery of the body, our blood, more specifically the water in our blood, would seep out of our capillaries, and very quickly our blood volume would decrease. Our blood pressure would drop, and we'd lose our ability to transport things throughout the body. So it's important that we maintain a certain amount of water in our plasma, and albumin is what does that. It grabs onto water. Albumin cannot leak out of the capillaries, and therefore the water cannot, or at least not to a great extent, leak out of the capillaries. So let's real quickly talk about what we just went over. So remember, the hematocrit is the proportion of red blood cells in whole blood, and it is the oxygen carrying portion of the blood. It's the gas carrying portion of the blood. Without hematocrit, oxygen cannot be delivered to our tissues. We'll talk about later how carbon dioxide does have an ability to actually be stored in the blood itself and not the red blood cells to some extent, but oxygen, on the other hand, could not be transported without hematocrit, without red blood cells. So red blood cells are very important in gas transport, and that's what the hematocrit is. It's the proportion of whole blood that is comprised of red blood cells, which are also called erythrocytes. Normally, it comprises about 45% of whole blood. So if someone's blood doping, or if someone's very dehydrated, or if someone is anemic, that percent might change. But typically in a healthy adult, it comprises about 45% of whole blood. All right, what are two protective functions of blood? We know that it carries white blood cells and antibodies, so it plays a role in fighting infection and protecting our body from invaders like bacteria and parasites. And we also know that it is able to reduce blood loss in the event of a rupture in some blood vessel. So if a small artery for some reason ruptures or is cut or severed, blood will clot and prevent further blood loss or reduce further blood loss. Very handy. All right, again, last one. Are plasma proteins used as fuel for body cells? Well, we only really talked about albumin, but I'll tell you guys as a general rule, protein in blood is not a fuel source. Cells need ATP to function or to undergo metabolism to carry out all their different protein productions and mitosis and things like that. They require ATP. ATP is metabolized from glucose and other derivatives of glucose. Glucose can be made from proteins, but that does not occur in the blood. Proteins and fats can be converted to glucose. That happens elsewhere in the body. The proteins in our blood plasma are not used directly for fuel for our body cells. The bulk of them are used for the retention of water in the blood to create that osmotic gradient, to keep the water from seeping out of our arteries into our body's tissues. And the one that we specifically talked about again was albumin. Albumin prevents loss of water from our capillaries into our tissues. All right, so we talked about plasma a bit. Now we're going to talk about some of the formed elements that are floating around in this plasma. Now, these are different from things dissolved in plasma like hormones, proteins, ions, things like that. These are actual formed elements. These are things that float around in the plasma. And there's really three basic categories: erythrocytes, leukocytes, and platelets. Erythrocytes are red blood cells, leukocytes are white blood cells, and platelets are fragments of much larger cells found in our bone marrow that are very important in the clotting process. Two of these are not even true cells, and I think you could probably figure that out pretty quickly, that platelets are fragments of cells, so they're not really true cells. And red blood cells do not have a nucleus or other organelles and do not undergo mitosis. They're really just bags of a protein called hemoglobin, so they're not really true cells either. The only formed elements that are true cells are the white blood cells or the leukocytes. Most of these do not survive very long in circulation. Again, red blood cells have no organelles, neither do platelets, so they're going to degrade or be used or removed fairly quickly. And then same with white blood cells. White blood cells can divide, and they do have organelles, but they move in and out of circulation readily. They typically do not stay in circulation for their entire life. Again, most blood cells do not divide. And that's true for, I don't want to say all, but that's true for most. Remember, most blood cells are produced in bone marrow, so they have no need to divide. These white blood cells here, for the most part, are not going to divide because they're produced in the marrow. Once they live out their life, they're removed from our body and degraded. Red blood cells, same thing. Platelets, same thing. All right, let's go ahead and get into the first of the formed elements, the erythrocytes. Now, erythrocytes, again, no nucleus or organelle, so they're shaped like a little donut. It's called a biconcave disc. It gives the red blood cell a lot of surface area, which is important in terms of if your job is to absorb a gas, you want lots of surface area. Also, it allows, what it does is it makes no one region of the cytoplasm too excessively far away from the cell membrane. If it was a sphere, then you could see that the center of the cell would be pretty far from the cell membrane. Because they're flattened, the center of the cell is actually now pretty close to the membrane. So again, it gives easy access to the cytoplasm. These cells are highly deformable. That's important. They can kind of stretch and squeeze in between the little capillaries of the body. And they are, like I said earlier, pretty much mostly hemoglobin, 97% hemoglobin. Another important feature of red blood cells is that they generate ATP anaerobically. Their job is to carry oxygen, so it makes sense that they do not use oxygen during metabolism or during the production of ATP. If they did, they'd be consuming the gas that they're trying to transport. Let's talk a little bit about a protein that allows red blood cells to do what they do. So, red blood cells, the primary function of red blood cells is to transport gas, specifically oxygen from the lungs to the body's tissues, and to a lesser degree it transports carbon dioxide from the body's tissues to the lungs for removal. The protein that allows this to happen is hemoglobin. Hemoglobin is made up of globin proteins and a group called a heme group, and that's in the blue here on the left. This is the heme group, and then these coiled proteins here, these amino acid chains, are globin proteins. The heme group, in each heme group, there's an iron molecule. Each iron molecule can bind to one oxygen molecule. Okay, so there's 4 heme molecules total. Each one binds to one oxygen molecule. That means that each hemoglobin molecule can bind to 4 oxygen molecules. There's roughly 250 million of these hemoglobin molecules inside of a red blood cell. So each red blood cell can transport about 1 billion molecules of oxygen. It's pretty incredible. When the red blood cell travels to the lungs, to the alveoli of the lungs to receive oxygen, and oxygen binds to the heme group, it becomes oxyhemoglobin and is bright red. As oxygen is removed and delivered to the body's tissues, it becomes deoxyhemoglobin, at which point it becomes very attracted to carbon dioxide, or at least more attracted to carbon dioxide than it normally is, and carbon dioxide binds to the globin proteins. It's important to know that carbon dioxide does not bind to the heme group like oxygen, but rather it binds to the globin proteins to be transported back to the lungs for removal. Blood cells are produced in red marrow, and this process is called hematopoiesis. About 100 billion cells per day are produced by the red marrow. It's pretty incredible. And all blood cells arise from a specific type of stem cell called a hematopoietic stem cell. This stem cell divides and can give rise to many different types of blood cells. Those blood cells themselves do not divide to reproduce new blood cells, but rather the stem cell divides to produce new blood cells. Specifically, if we're talking about erythrocytes, the process is called erythropoiesis. Again, this occurs in the red marrow. A stem cell is going to undergo some type of change in which it becomes a committed cell, meaning that there's no turning back. This cell is now going to differentiate into its end product. This committed cell goes through 3 steps, and there's a key factor in each of these 3 steps I want you guys to know. In phase 1, the early erythroblast, which just means a very young erythrocyte, undergoes ribosome synthesis. Remember, ribosomes make protein, and red blood cells are packed with hemoglobin, which is, for the most part, a protein molecule. So it needs lots of ribosomes. Those ribosomes begin to produce hemoglobin in phase 2. And during phase 2, hemoglobin is produced. At the beginning of phase 3, the nucleus and organelles are ejected from the nucleus, at which point it becomes a reticulocyte. A reticulocyte is just a young erythrocyte that has either just getting ready to enter circulation or has just entered circulation, only for a small amount of time. This whole process takes about 15 days from start to finish. And in healthy people, reticulocytes account for 1 to 2% of total erythrocytes in our blood. If this number is higher, it means we are working harder to produce new blood cells. If this number is lower, it means we are not producing enough red blood cells. So someone may have a high reticulocyte production rate, meaning they may have 3 or 4% of their total red blood cell count is reticulocytes. That could mean they had recent blood loss. That can mean they're destroying red blood cells too quickly, things like that. If you go give blood, most likely after giving blood, you'll have a higher proportion of reticulocytes than normal in your blood. All right, the rate at which red blood cells produced is regulated primarily by your kidney. So on average, about 2 million erythrocytes are produced per second, but this process can be increased or decreased, and it's controlled hormonally. Basically, kidneys detect reduced oxygen in the blood. They detect hypoxia. In response to less oxygen being detected by the kidneys in the blood, they secrete a hormone called erythropoietin. Erythropoietin is the hormone that stimulates increased erythropoiesis, or increased production of red blood cells, and it acts on the bone marrow, on the red bone marrow. So the bone marrow then, in response to erythropoietin released by the kidneys, begins to produce more erythrocytes and therefore increases the oxygen carrying capacity of blood. This process is controlled by a negative feedback cycle. So as increased oxygen reaches the kidneys, the stimulus that triggered this event in the first place is now removed. Remember, the stimulus was hypoxia, or decreased oxygen. So now that we have more oxygen, erythropoietin ceases to be released, and our the production of our red blood cells by our red marrow goes back to normal. It goes back to the typical 2 million, give or take, rate. And again, in response to low oxygen, this cycle can occur again, can re-stimulate. So if you give a liter of blood today, erythropoietin is released until your oxygen carrying capacity of your blood is restored to normal, and everything goes back to normal. If you give blood again the next day, this process repeats itself. So it's just a hormonal control based on a negative feedback cycle that controls the oxygen carrying capacity of our blood at any given time. Figure 17.7 in your book. The only thing that really adds is it talks about how red blood cells are destroyed. Basically, they lose their ability to deform easily, and they get stuck in the very small arteries of our spleen, as well as other places, at which point they are phagocytized and removed from circulation by white blood cells. All right, there are 2 general categories of erythrocyte disorder. One is essentially a condition when the blood is unable to carry as much oxygen as normal. It can't support your body's normal function because it cannot carry enough oxygen. So this is a sign of some issue going on, of some other problem in your body. It could be blood loss. It could be that your body's not producing enough cells, or it could be that too many cells are being destroyed. 2 examples your book gives are thalassemia and sickle cell anemia, 2 conditions in which a defect in hemoglobin causes cells to form incorrectly, causes red blood cells to form incorrectly, at which point they are destroyed. Polycythemia is a condition, sorry, I am still sick, sorry guys. Polycythemia basically means many cells, and it's an abnormal excess of erythrocytes that makes blood too viscous. One example you may hear about in the news is blood doping. This is when you remove your blood at one point, you get the hematocrit out of it, and then prior to a competition or a race, you put that blood back in your body to increase the number of red blood cells in circulation, to give your blood a higher oxygen carrying capacity than normal. Now, this can be bad because obviously you can imagine if you start to stuff a lot extra formed elements, red blood cells, artificially into your blood's going to become more viscous. All right, the ratio of plasma to red blood cells is going to change, and your blood is going to be more viscous. If you combine this with dehydration, it gets even worse. So you can see how in a big athletic event where you're sweating a lot, this could become a bit of a problem. Polycythemia vera, this is a bone marrow cancer in which you produce very high amounts of red blood cells. I'm reading here in the book that with this condition, your hematocrit can be as high as 80% of blood volume, or of whole blood, sorry. So you can see how this is a bit of a problem, right? If 80% of your blood is red blood cells, it's going to become less fluid. It's going to become highly viscous, and it's actually going to increase your blood volume, which can be a problem as well. Just a quick question on what we just covered. How many molecules of oxygen can each hemoglobin molecule transport? Remember, there are four heme groups bound to the globin proteins in a hemoglobin molecule. Okay, so 4 heme groups per hemoglobin molecule. Each one of them can bind to one oxygen molecule. That means that one hemoglobin molecule can carry 4 oxygen molecules. And I kind of already answered the second part of this question. Which part of hemoglobin binds to oxygen? The heme group binds to oxygen. Very specifically, it's the iron molecule at the center of the heme group that binds to oxygen. All right, white blood cells. So white blood cells are not nearly as common as red blood cells. There's only about roughly 11,000 of them, under 11,000 of them. If you have more than 11,000 leukocytes, this is leukocytosis. This is a problem of having too many blood cells, too many white blood cells, sorry. In normal whole blood and healthy whole blood, leukocytes and platelets combined will account for less than 1% less than 1% of whole blood. Sorry. There's 2 general types of white blood cells, granulocytes and agranulocytes. These are named for their appearance under the microscope when they're stained, when they're dyed. Granulocytes take on a granular appearance, as you can see in the eosinophils, basophils, and neutrophils. The agranulocytes, the lymphocytes, and monocytes do not appear granular because they do not react to the stain the same way. Granulocytes first. Okay, so neutrophils account for about 50 to 70% of the white blood cells. They are the most common, and they become very common in response to a bacterial infection. Neutrophils destroy bacteria. That's their job. They eat the bacteria, phagocytize it. They can also release bleach and hydrogen peroxide to kill bacteria, which is referred to as a respiratory burst. And they even can develop spears that stick out of their cell membranes to puncture the cell membranes of bacteria. Pretty exciting. It's kind of crazy to think about these little guys. Eosinophils are 2 to 4% of white blood cells. They are going to go after parasitic worms. So worms are big, obviously, in relation to a cell. So our little cells cannot phagocytize these worms. So instead, eosinophils are going to release chemicals that kill the worms. They will gather around the worms and then release these chemicals. Basophils are relatively rare. They comprise about 0.5 to 1% of white blood cells. They're going to release histamine to help mediate our inflammatory response in response to an infection, an allergic reaction, a cut, things like that. All right, 2 types of agranulocytes. Lymphocytes are fairly common. They comprise 25% of white blood cells, but they are not commonly found in blood. They're very rare in blood. They reside instead in our lymph vessels, our lymph system. Lymphocytes play a variety of roles in our immune response. They can actually produce antibodies. that go after and circulate throughout our body to go after invaders. And then other types of lymphocytes can mark the invaders, they can destroy the invaders. So we'll talk more about that stuff in the immunity chapter, in the immune system chapter. Monocytes, on the other hand, these actually are not active in the blood, but they can circulate around in the blood until they get to the area of interest, the area where they need to be, and then they leave the blood and become macrophages. And macrophages can move easily throughout our connective tissues of our body and phagocytize invaders. 2 leukocyte disorders. I can't remember if we talked about these in class or not, so I don't think I'll go into it too much. Let's see if there's anything I want you to know. No, it's okay. It's on page 645 if you're just curious. I think mono is kind of interesting if you read the section infectious mononucleosis, as well as leukemias. Leukemias are very devastating. So actually, I'm going to go ahead and read it, but it won't be on the test. So leukemia, which literally means white blood, refers to a group of cancerous conditions including overproduction of white blood cells. As a rule, the renegade leukocytes are members of a single clone, okay, so they're descendants of a single cell that remain unspecialized and proliferate out of control, impairing normal red bone marrow function. The leukemias are named according to the cell type primarily involved. For example, myeloid leukemia involve myeloblast descendants, whereas lymphocytic leukemia involve lymphocytes. The more serious acute forms of leukemia affect children primarily, but also the chronic types occur very often in elderly people. All leukemias are fatal without therapy, and only the time of their course differs. So leukemia is pretty bad news. Basically, when you have leukemia, these defective leukocytes fill your red bone marrow cavities, and these immature white blood cells flow out into the bloodstream. You can imagine if white blood cells are filling your marrow and flooding into your bloodstreams, your other blood cell types are going to be basically crowded out and underproduced. So the primary effect of leukemia is going to be severe anemia and bleeding problems as well, because you're not going to have normal functions of your platelets. So to make it really basic, leukemia is essentially an overproduction of white blood cells, so much so that your body is not able to produce the other formed elements adequately. So you're not going to be able to produce enough red blood cells or platelets. And we know what both of those things do. Both of those things have very important roles, so you'll become likely anemic and have issues with clotting. Okay, I didn't mean to do that, but it's interesting to me, so there you go. Platelets are the last formed element that we're going to talk about. They're fragments of very large cells called megakaryocytes. They're actually really fascinating. There's a paragraph in your book here I want to read because it's so interesting to me. Okay, here we go. So basically after platelets are formed, and we're going to come back and talk about this formation pathway in a second, but after the megakaryocyte forms, again, and this is going to develop from a stem cell, just like our red blood cells and white blood cells. After the megakaryocyte forms, it's going to press against the sinusoid, and this is a type of capillary in the red marrow. So basically, it's going to press against the capillary in the red marrow and send extensions of its cytoplasm through the wall into the bloodstream, which is really interesting, right? So these little fingers of the megakaryocyte are sticking into our bloodstream, into our capillaries. These little fingers, these little extensions are then going to rupture and break off, releasing the platelet fragments into the blood. Those fragments are then going to quickly seal up. So it's a little piece, like a little stamp or something, a little piece that breaks off of the megakaryocyte. It's then quickly going to seal up. So it's a little tiny little bit of cytoplasm bound by cell membrane. And they're tiny. They're 2 to 4 micrometers. and they are actually surprisingly common. So each microliter of blood contains roughly 150,000 to 400,000 tiny platelets. Someone who is prone to clots, obviously, will probably be given a medication to inhibit platelets. And if you hear about drugs like thrombolytics, anticoagulants, those are all going to have some type of effect on platelets. And we'll look at what exactly platelets do here on the next slide. So platelets are really highly involved in the clotting process, which is referred to as hemostasis. There's 3 steps in the clotting process. Step 1, vascular spasm. 2, platelet plug formation. 3, coagulation. So step 1, basically, you get a tear in, let's say, an artery. Step 1 is for that artery to reduce the amount of blood gushing out of the tear. So what it's going to do is it's going to constrict. It's going to try to close down. So blood is not rapidly being lost from the artery. This buys time for the artery to repair itself. Step 2, platelets are going to adhere to the exposed collagen fibers of the tear. When in solution, platelets don't normally like to bind to each other, which is good because you don't want platelets binding together and bunching up in circulation. But when they're exposed to collagen, they like to bind to each other. They like to adhere to each other, and they like to adhere to collagen and form this little platelet plug. The platelets are then going to release chemicals that make all the other platelets around them sticky as well. And this is just going to build and build this platelet plug to slow blood loss. After that happens, fibrin is going to form a mesh around these platelets that forms a little, almost like a net. And whatever blood is still leaking out of this rupture in the vessel, many red blood cells will begin to get caught in this mesh, in this net, and they'll ultimately seal the plug and coagulate and make a little fill in this tear so we stop losing blood. Then over time, this clot will be repaired and this tear in the vessel will be repaired. But this is just the immediate way to stop continued blood loss. Okay, I tried to hurry through that to get to this kind of, not that it's any more important than any of the other portions of the book, but I think it's kind of difficult to understand. So I want to talk about transfusion and blood replacement, especially because it seemed like this was what we were struggling with the most last night, myself included. So let me just take a second to walk you guys through it again. We're going to talk about blood types and how the different blood types react with one another. There's a few different types of antigens, which remember are markers that are found on red blood cell plasma membranes. Okay, they're highly specific glycoproteins found on the cell membranes of red blood cells. These specialized type of antigens on red blood cells are called agglutinogens. There's 30 groups of these agglutinogens, but ABO and RH are the most important. And we're really just going to focus on ABO, actually. I'm just going to go ahead and say, don't worry too much about RH. It's really the same concept, but let's just focus on ABO and master that, okay? All right, so what your blood type is referred to, what I mean when I say you're a type A or a type B blood type, this naming convention is based on the presence or the absence of 2 types of agglutinogens. And from now on, I'm going to say antigen, but what I really refer to in this instance, it's really an agglutinogen. Okay, so there's type A antigens and type B antigens. That means there's blood cells with type A markers on them, type B markers on them. They may have type A and B markers on them, or they might not have any markers on them. And all the different types of blood are going to be named based on whether or not they have these, they have some combination of these, or they don't have either of these. Okay? I'm going to come back to the agglutinins, but I want to go to this slide right here. Okay, so let's just look at antigens for now. Type A blood has type A antigens on the red blood cell. See that? Type B blood has type B antigens on the red blood cell. Type AB blood has A and B antigens on the surface of the red blood cell. And type O blood has no antigens on the red blood cell. Okay, so we know the antigens. Now what we need to know to figure out which type of blood will react to which type of blood is what antibodies are present in each. Okay, so these little blue things are antibodies, these are antibodies, these are antibodies. Now this is important because if you look at this little picture down here below, we see that antibodies can react with antigens and cause clumping or agglutination, which is bad. It can cause renal failure and a lot of cell death. So, certain antibodies are going to react with certain antigens, okay? And this picture is really helpful because it shows you in blue that these blue antibodies are going to react with these blue antigens, and these red antibodies are going to react with these red antigens. So let's just take a look at type A and B blood first. All right. So type A blood, again, has type A antigens, but it has anti-B antibodies. So if it finds B antigens, like the B antigens present in type B blood, they're going to destroy it. All right. So now, if I give type O blood to this type A person, these anti-B antibodies will not react with the type O red blood cells. Do you see that? Because there's no B antigens. These anti-B antibodies will not react with type O blood cells because there are no type B antigens. Now here's what is going to be tricky, and I'm going to ask a couple questions on the test like this. You have to remember that it is the recipient's antibodies that react with the donor's antigens. So the recipient here has anti-B antibodies. So if I give them B antigens, donor antigens, if I give type B blood to type A blood, the recipient's antibodies will react with the donor's antigens. Okay? Now, if I give type O blood to this type A person, these anti-B antibodies will not react with the type O red blood cells. Do you see that? Because there's no B antigens. These anti-B antibodies will not react with type O blood cells because there are no type B antigens. Now here's what is going to be tricky, and I'm going to ask a couple questions on the test like this. You have to remember that it is the recipient's antibodies that react with the donor's antigens. So the recipient here has anti-B antibodies. So if I give them B antigens, donor antigens, if I give type B blood to type A blood, the recipient's antibodies will react with the donor's antigens. Okay? Type AB, all right? Type AB is known as the universal recipient, meaning that they can accept any other blood type. They can accept type A blood, they can accept type B blood, and they can accept type O blood. And that's because they have no antibodies. They have no antibodies that will attack the antigens on any of these cell types. You should see something that kind of makes you take pause and, well, how does this work? And the problem is that when I give a type AB, remember, type AB is a universal recipient. When I give them type O blood, sure, there's no antibodies in the recipient to attack the donor's antigens, but what about the donor's antibodies? Won't the donor's antibodies attack the recipient's antigens? And remember, in agglutination, it's the recipient's antibodies that attack the donor's antigens, okay? That's what we need to worry about. But you might wonder, well, what about the donor's antibodies? Aren't they going to do something to the recipient's antigens? The answer is yes. These antibodies will attack these antigens, but there aren't enough antibodies being donated to really have a severe reaction, okay? So if I give type O blood person to a type AB person, then yes, there will be some minor agglutination that will occur between the donor's antibodies and the recipient's antigens. But remember, you have to remember, severe reactions are caused by the recipient's antibodies. Remember, type AB blood has no antibodies, so the recipient's antibodies in the donor's antigens. So this would be an okay reaction. I could give type O to type AB blood because the recipient does not have any antibodies to attack the donor's antigens. And in this case, the donor doesn't have any antigens. So it's a win-win, all right? So that hopefully should explain a little bit why type AB blood is the universal recipient, because it has no antibodies to attack the donor's blood, and why type O blood is the universal donor, because it has no antigens to be attacked. Okay, so if you have to write down, severe reactions are caused by the recipient's antibodies attacking the donor's antigens. That's the problem, okay? The recipient's antibodies attacking the donor's antigens. So O blood can be given to AB blood. What if I switch that around? What if I give type AB blood to a type O person? So I donate type AB blood, and I put it inside of a type O person. Well, the type O antibodies, the recipient's antibodies, would attack the donor's antigens. That's a problem, okay? All that to say this. On the test, what I'm going to do is phrase a question, probably a handful of questions, asking if severe, possibly fatal agglutination will occur in this scenario. So, if I give type O blood to a type AB person, will severe, fatal agglutination occur? The answer is no, because the recipient's antibodies will not attack the donor's antigens. Remember, that's a fatal reaction when the recipient's antibodies attack the donor's antigens. Another question. What if I give type AB blood to a type O person? Will potentially fatal agglutination occur? The answer is yes, because the recipient's antibodies will be attacking the donor's antigens. So if you get anything out of that, replay this little part right here. Be sure if I ask you, will this blood react with this blood, you'll have an answer for me. Let's do a couple more examples. What if I give type A blood to a type B person? Well, the anti-A antibodies in the recipient in the type B blood will react with the type A antigens. So yes, there will be a severe, possibly fatal agglutination. What if I give type B blood to a type A person? Well, the anti-B antibodies in the recipient will react with the type B antigens in the donor. Severe agglutination will occur. If left untreated, death could occur. Okay, what if I give type O blood to a type B person? The anti-A antibodies in the recipient have no antigen to react with. So severe agglutination will not occur. And again, remember, type O cells have no antigen, so they're the universal donor. What if I give type A blood to a type AB person? Well, there are no antibodies in the recipient to react with the donor's antigens, so severe blood agglutination will not occur. Remember, it's the recipient's antibodies that have to attack the donor's antigens in order for severe agglutination to occur. Okay, if you guys have any questions, please email me, text me, call me, whatever, and we can go through that again. Anytime mismatched blood is transfused, for example, if I give type A blood to a type B person, the agglutinins, or the antibodies, will attack the antigens of the donor blood cells, okay? And based on what we just learned, Group O is a universal donor because it has no antigen markers present, and Group AB is a universal recipient because it has no antibodies present. All right, guys, hopefully that was helpful. I'm going to get to work on the study guide right now. Please let me know if you have questions. If you don't think you'll be able to answer questions like, could a potentially fatal reaction occur if I give a type A blood person type B blood? If you don't feel like you can answer that, make sure you call me, get in touch with me, and we talk about it, okay? All right, you guys, take care. Don't hesitate to ask me for help, and I will see you on hopefully Wednesday, but worst case, I'll see you on Monday.